One more thanks to our keynote speakers. Russ and Larry both gave phenomenal talks, and we're really appreciative that they both spent the time to do that. I think it was long overdue that we had the two of you speak here at PSB. It was really a treat for all of us, so thank you to both of you. I don't know if Russ is in here right now, but I see Larry, so thank you. One last thanks to all of the funding, all of the PSB staff. They have done a phenomenal job all week behind the scenes, making sure that everything happens. All of the co-chairs, everything that you've seen has been a lot of work by a small group of people. And also thanks to all of our sponsors. They enabled us to have the quality of food that we were able to have, having drinks at the poster session, things like that are because of the sponsorship. And so this is my shameless plug that I'm already looking for 2020 sponsors. So if you're interested, please send me an email. If you've sponsored before, I will probably reach out to you in just a few months. So keep that in mind. And so lastly, thank all of you for participating, for coming to Hawaii. Without people coming to this meeting, we wouldn't have a conference. We hope you enjoyed it. We're very much looking forward to the 25th next year. The organizers met yesterday and started talking about what we might want to do different and fun for the 25th anniversary of the conference, and so it'll be an exciting conference for sure. So have a safe journey home, and mahalo and aloha. So thank you. All right. Now I'm going to introduce our first Terry talk for this morning. Graciela Gonzales is going to give a talk on social media for health research and dispel some of the myths around that area. She's an associate professor of informatics in the Department of Biostatistics, Epidemiology, and Informatics at the University of Pennsylvania Perelman School of Medicine. So she is one of my colleagues. We're both also in the Institute for Biomedical Informatics. She came to Penn about almost 2.5 years ago already. She had spent 11 years in Arizona State University before that. She's an expert in NLP and medical and clinical informatics and health informatics, and we're delighted that she was willing to give a Terry talk. So thank you, Graciela.
Thank you. Thanks, Marilyn. Thank you. All right, good morning. Thank you for being here up to the last day. So I'm delighted to give this Terry talk. It was intriguing and trying to choose a topic and on what to talk about. And I decided that I needed to talk about something that I'm passionate about. And I'm definitely passionate about my research. And so I opted for the research route in this case. But otherwise, there's that little tiny URL that has the unofficial bio that I told at Penn, and they published it in the online newspaper that they have. So if anybody's curious about all those little juicy details of my past, some of them are there. So anyhow, my research in social media for health has been sponsored by the NLM for the past 7 years. So my grant just got renewed, my R1 just got renewed on it. So along the way, we've learned many, many lessons that I would like to share some of them with you. So this is my opportunity to actually lecture about the things that bug me along the way when I present my research. So I guess in that sense, I'm thankful to Terry for inviting me and having me talk in this format. Okay, so they told me up is the pointer, down is the other one, but none work. To switch, okay, I'll go with enter. So social media, we want to use it for health research, and basically what we're hoping to do is get these groups out of social media, these cohorts that then we can study. So in order to do that, there's like many traps, this. And so if you look closely at this image, I don't know if it's clear from there, does anybody notice anything strange about it? Duplicates. Yep, that group and this group are the same. So my little circles are pointing out this doctored image that you see here with many, so a crowd was made to look bigger right there. So in essence, it kind of summarizes some of the things that we're going to look at. How to choose how to distinguish different groups in social media and some of the myths that exist about how much data you can get and how can you use it. And of course, on the sound scientific principles that you need to follow in order to get good studies out of social media data. So, first myth, nobody will say that on social media. And by that, this is the most broad variable that I can find for this. It's like, you just name it, put it there. Every single thing that we've tried doing or that we've done, and I bet everybody that does social media research gets that first. And it was funny because that was the first thing I heard after the workshop where I gave a little bit more detailed talk about some of the work we're doing. So yeah, I've come to believe that everything or anything that you say, it's in social media. One way or another, be it Reddit, Twitter, Facebook, Instagram, it's there, okay? So we have made some advances in finding it. This is a very simple one. I gave one like this in the last talk, but it's not the same search. So if you try it, I've been diagnosed with prayers here brings down the level of noise a little bit. So you get a bunch of the real announcements about being diagnosed with all sorts of diseases. Some of the history or some of the things that they mention are very specific about what happened and how it was. And I chose this one because later on I'm going to talk about using common sense when you show social media data, because specifically this person seems to be aware that he's making it public. He says, my wife and my family know. For the past few weeks I've had very low energy. After some blood tests and conversations with my excellent GP, I've been diagnosed with depression. And then continues, my wife and family know, my church and colleagues know, I figure I tweet about it, so please keep me in your love and prayers. So this person seems to be aware that that tweet is public and is meant to be public. So that's one of the other ones, the other gray areas, the other things that we deal with when using social media data. And so if you try that, you'll get everything from cancer, HIV, anything that you think of comes there. At some point, I considered writing a paper titled like that. I've been diagnosed and other social media confessions, but I mean, the rest of the work took over, so I have never done it. So maybe one of you can pick up on that idea. So what we did is we went with trying to study pregnant women. So this paper was published in the Journal of Medical Internet Research. This was our first paper in a series. This one was where we basically were discussing how we found our cohort of pregnant women, which right now should be around 150,000 of them after classification and actually clearing a total of about twice that. So from 300,000 timelines, about 150,000 will be timelines of actual pregnant women, and leaving out bots as well. Bots are pregnant sometimes, or appear to be, anyway. With that, and this is the simplest of NLP, a word cloud. This is from some of the timelines in that cohort, and of course something is going to jump at you. It's like medication. Tylenol seems to be the most popular one. And we see some of the tribulations: pain, sleep, sore around, headache. Husband is over here somewhere, small. And we get a full picture. I don't know if by looking at the picture you could tell it's from pregnant women, but that's not the point of doing this kind of exercises. So the language exercise, or the pure fun of using language and language techniques, is not what drives this research. Which leads me to myth number 2. Okay, you can collect tons of data from social media, and this is like the colloquial use of this. I see this in research proposals. So whenever I get research, I get, of course, the scientific equivalent of that. Like, we're going to collect 20,000 of this in 3 months or less. And so because you're told that you have to be quantitative, so they put 20,000, 30,000, 50,000 with no basis whatsoever for it. So the first thing I look at when I'm reviewing proposals that involve social media, and I get a bunch of them, is what is their search strategy? How are they going to actually get this data? Do they have a solid, sound approach to obtaining this data? Particularly anything that are rare events. So that's the other temptation, thinking that you can get anything, which is myth number one. Once you stop believing that you cannot get it, then you go to the other extreme and think you can get everything and everything. So the fact that it's there doesn't mean that you can get it. At least not easy. So the collection approach, the collection mechanism is very, very important. We have this paper recently published that is solely dedicated to actually how to get some of these rare events. Birth defects in general, they account for about 3% of pregnancies result in a birth of a baby with some sort of birth defect. So if you think about it, out of our collection of 150,000 women, we could only expect about 3% of them to have pregnancies that end in a baby that has a birth defect. So in and of itself, the reality of it is already reduced. And in general, in social media, you don't get a true reflection of what goes on in the general population. You get some of it. So in the end, and without meaning to spoil the reading and enjoyment of this paper, we got about 1.4%. So finding 1.4% of timelines in such a large cohort is a very, very interesting challenge for informatics and informatics methods. So it is not easy peasy to find things. So here I just meant to walk you through this briefly. So if you see this top part, those are all the steps to get the initial query formulation that would allow us to do our work or to actually approach some, I was going to say computable, but processable set of tweets among the many millions that we had. So we start with a lexicon. We get lexical variance, which for social media means not only synonyms and so on, but it means misspellings of that, popular names for it. It means misspellings of the popular names for it, and other expressions that we haven't even heard about, and that definitely will not be in the lexicon, but we need to start somewhere. So we got regular expressions, and we get regular expressions that will attempt to capture. In this case, we sent them to the Twitter pregnancy database, or we submitted it against our pregnancy database collection, got the matching tweets, started post-processing to reduce some of the noise. A lot of rules and programs that reduce noise will help here. And then we look at frequency distributions, meaning we look at some of the regular expressions that yield a high number of tweets, those that do not yield anything, and then why. And then after that, extensive manual analysis will give us extra things to add. So actually this will probably come over here more than anything. But we learn from the data, but the way we learn at the beginning of a problem is all manual, okay? So it's all due to somebody taking very careful steps to get and extract more expressions, try them, put them as regular expressions, and then circle here until you get something that you can deal with for annotation. So at the beginning, so this is the first steps, okay? So we got text annotation, and with this we get, what we wanted to do is get a positive for birth defect, and then there is some maybe, because the tweet itself doesn't give you enough information, we wanted reports, first-hand reports, meaning the mother of the child is reporting, because we were interested in pregnancy timelines. It's like what happens during pregnancy that leads or that may have some hints about the origins of the birth defect. So sometimes the tweet doesn't give enough information. So what we do in that case is we just mark it as maybe. That's the question mark. And others that are definitely negative. So this could be concerns about birth defects, for example, which if you're just searching for birth defect mentions, then you'll get those, a lot of them. And then after that, we get, from the positives, we still need to find the time frame. So we needed not only that they have a child with a birth defect, but that pregnancy was within the timeline that we could collect. And so we had finally our birth defect cases, okay? So that's just our first step, because after that, in order to do any study that is worth its dough, is you need to go on and continue with what I'm gonna mention next as the next myth. So we'll hang in there. We got you to the cases. So just to summarize, if that it is there doesn't mean that you can find it. That you can, and I keep with this, that you can find it doesn't mean a program can find it. The Twitter graphical user interface is different than the application programmer interface. So the API doesn't necessarily do the same thing as the graphical user interface. For example, it doesn't take patterns. It only takes keywords. So that little pattern about I've been diagnosed with will be turned into diagnosed with and I've together. So 3 keywords rather than 1. And they could occur anywhere in the tweet. And then after you collect like that, then you have to go and do Lucene indexing and pattern and so on. So it's not a straightforward shot to go from a graphical user interface to what you can do in a program. Plus, now Twitter is tightening their application programmer interface licenses, so it's hard to get one now, or harder to get one. That a program can find it doesn't mean that that information that you get back is valid for your study. So as I said, we got a lot of the mentions of birth defects, but that doesn't mean that it is a mention of the baby having a birth defect. And like that, medication name. Well, all the mentions of medication name doesn't necessarily mean that it's like someone taking that medication. So we have to write the classifiers to distinguish the 2 classes. And that's at the very beginning of all this. And validating then requires annotation, extensive annotation. So getting a cohort, so next time you find this nice set of tweets that have been annotated, grab them and use them. Because getting to that takes a lot of effort and a lot of time that you may not have for your research, particularly PhD students. So we're making available a lot of these social media collections of tweets. Then we have, okay, so scalable annotation requires classification. I just mentioned that. So in order to be able to give something to our annotators that is doable, you need to do some classification up front. And in order to do some classification, you need to do some annotations. It's kind of a little conundrum there that you need to get out of quickly. So misconception number 1, that you only need to analyze what you're interested in observing. So, oh, I found my cases, I found my birth defects, so that's it, just like, forget it. Or also known as, you don't need controls. In social media studies, it's an observational studies, just like any other observational study. You need both, you need cases and you need controls. And finding the controls sometimes is harder than finding the cases. Because in this case, for example, for the pregnancy data, it meant, well, how do we find timelines of women that gave birth to a child that's healthy? Easier said than done, because out of the collection, we needed to think about all the possible things that could go wrong and rule them out, or definitely find somebody mentioning that the baby girl or boy has been born healthy and so on and so forth. So we first go to that, and then we take other measures to try to get more occurrences. There's not too many people that specifically say this. But here's where the volume of data in social media helps. Sometimes we do get a lot or enough, at least to start our study, with just the very precise queries that will give us exactly what we want. So this is the step 2 of that first paper that I show you. This is the one where we actually went and did a pharmacoepidemiologic evaluation of the birth defects data that we had collected. So here is where we look at which birth defects were mentioned and what frequency and so on, and we needed to find the denominator, which again threw us into trying to find out which of the timelines were actually pregnancy timelines, which threw us into finding the bots, because there's bots that repeat these pregnancy announcements a lot. So we needed to calculate, so that threw us into trying to find a pregnancy time frame as well, automatically from the data. So meaning, when did the pregnancy start, when did the pregnancy end, do we have tweets for that period, and now do we have a due date? So finding all these little pieces, and they translate, well, it's like, yeah, one of the comments of the reviewers is like, well, this is a very specific problem. Like, yes, you have to solve the very specific problems in order to actually do anything that's health related. And that's the problem, and a separation between computer science and biomedical informatics comes into play. We send these things to ACL and EMNLP and so on, and they don't care that it's about pregnancy and health and what we can get from this. That's not their purview. They want, what are the NLP methods and magic that you did to get this thing? So here is where this defines the field in a way. Like, it's not so much as what tricks I do in NLP, which sometimes we do need tricks, but it's about how we're using the data, what impact is it going to have, how are we going to be shaping that into a study that then can shed some light into actual scientist questions, or questions that or domain experts have. So we're helping them get their questions. So this is a continuation of my prior diagram that gives you some of the numbers. So variance, regular expressions, this is what we got, 120,000 users, timelines at first. And of course, that's not going to be, you cannot annotate that easily. You have matching tweets and so on. Then after all this, after swirling around in that block at the top and refining it, we went from 119,000 and so on down to something much more manageable, which was 5,000 users and 16,000 tweets. At that point, and just to give you an idea, this meant about 150,000 tweets at the beginning, down to 100,000, down to 75,000, down to 50,000, 20,000. So by the time Ari Klein, who's the lead author in this, got to 16,000 tweets, he said, I quit. I'm going to annotate 16,000 tweets. I don't care. So he just went, I'm not refining my queries anymore. So he went with that, and then finally, after all the filters, we get to 196 users. From 119,000 to 196 after all the filtering. So, and before I go there, briefly then, so getting to this then requires a lot of combination of automatic and manual uses. Until we can nail the manual process, we cannot move on to do the automatic process. So myth number 3, research using social media is human subjects research, which means all these forms, and that means we cannot include it, we cannot do it. It's actually not, by definition of the NIH, of what constitutes human subjects research. Using publicly available social media data will not make it fall under as a human subject research. It is not. So you will be able to conduct it as long as you stay away from any data that's not publicly available. So if you join, that is like a broad thing. But for example, it means that the groups I join, say in Facebook, on my personal Facebook account, and it's a private closed group, I do not use it because it was not meant public, it was meant private. You still need IRB certificate of exemption. You still need to apply to your institution and get this exemption certified. You still need to abide by responsible conduct of research. You still need to use some common sense, okay? So it doesn't mean because it's public that, remember that tweet I put at the beginning, that you can just grab any tweet and just put it out there in slides or in papers and so on without regard to the person that tweeted it. you have to be very, very conscious of what was the intent, of what was what the person intended to do or expected of this tweet, and then some other things. So, for example, we found one tweet that mentioned from a pregnant woman that had a baby with a birth defect, but in her timeline said, sitting here in my car, chain smoking, finally, I'm tired of this not smoking thing, and I don't care. So basically, something like that. Of course, I'm not saying the exact words because the way it was worded, if you put it in Twitter, you will get the name of the woman. So what we did in that case is we slightly altered the tweet in order to publish it. So if it said the brand name of the car, for example, or the make of the car, we switched it. So we basically switched the language a little bit so it couldn't be found, and we tried it on Twitter to make sure that when we publish it, if, I don't know, that woman's mother-in-law happens to be in the audience, will not tweet it and find her name. I know she has enough in her life without having more family members find out about this instance that may or may not have caused the birth defect, so you still have to use that common sense. So, and then there comes a point when even if social media data is public, you have to know when to get consent. The people that you find, the good thing, people that you find on Twitter, you get their, you can private message them and invite them to join your study. So that's the thing that we're gonna be coming next. And I'm wrapping up. Misconception number 2 is that you cannot share social media data. And this is an excuse used by many researchers that are doing research and annotating the social media data and not sharing. So that's one of the things that gets me going, is like, you're like, okay, oh great, can I use your corpus? You're like, well, no, we cannot share social media data. Yes, you can. As long as you abide by the Twitter the Twitter terms of use, you can share social media data. You will share only the ID, and then people can pull the tweets, for example. You can share up to 50,000, for research, you can share up to 50,000 tweets openly, meaning without the Twitter ID. Myself, we choose to do just the Twitter ID, just to respect people that wanted to delete their tweets so they won't show up. For reproducibility, though, we need to make it available. So here is this really neat graphic that I did not create. This was from Renee Costers. From research integrity to scientific misconduct, there's a spectrum of things that you can do. And so I will always encourage data sharing, open code, and so on. So that's just the conclusion. Social media data can be valuable. It is abundant, but not easy to get. It's technically not human subject research, but you still have to be careful with what you do and how you do it. And its value will depend heavily on good research design and solid scientific practices. I'm going to stop there so I can have time to take some questions.
Graciela, I had a question. When you display individual tweets, do you seek out consent in those cases, like the one you showed here in your previous talks?
Oh, okay. But what's the question, though?
Consent of the ones you showed.
No, because as I explained, that tweet, first of all, you should not or you do not have to get consent for every single thing because it's publicly available data. So in that sense, legally I don't have to get it. And my common sense dictated that that tweet was meant to be public because he said so specifically. So I reduce a lot of the tweets that I show to a minimum to prove a point and not more. And I will not, for example, show you that real tweet that we found of this woman and so on. So again, it's a lot of common sense. Yes, Larry.
Graciela, it's lovely talk.
Thank you.
I want to follow up a little bit on your very last point about
Sure.
scientific method around this stuff. So observational studies generally have uncontrolled risk of error because it's hard to know about confounders.
Yes.
And you go through so much effort to try and find the right collection of tweets, and even though you're trying to identify controls, it seems to me that the risk of confounders is huge. So can you talk methodologically a little bit about what you can do to try to reduce that risk?
Sure. One of the things will be annotation. So in order to find our 200 controls, for example, we thought about what will be a good way to reduce the number of confounders. So we went with something that's very simple, and it actually didn't make a difference in the end, which was the same pregnancy time frame. So the controls match one-to-one with the cases on when the woman was pregnant. So we tried in that way. We tried to find, so since we're not establishing causality or claiming that this is caused by, at the point that we collect our controls, we basically do all the statistical studies on them and p-values on differences and so on that we find, and we stop there. So, for example, we knew which drugs were consumed more often by women that had a birth defects outcome, but we did not publish that because we thought that will be not solid research in that sense, and it could throw, and not that we are like defending pharmaceutical companies or anything like that, that's not my motivation. My motivation is how much reason do we have to list this drug that we found to be very common in, or more common in the cases versus the controls. I don't have any because the controls were also chosen somewhat, not randomly, but with not enough rigor to eliminate all the confounders.
Control for things like population prevalence of phenomena. So Twitter is not a representative sample.
Yeah. It's changing, but yeah, we know, we're aware of all that. And that's exactly some of the things that we get back when we send proposals. It's like, well, but our claim is that none of the collection mechanisms that are out there will have the same assurances. So nothing is perfect. And it doesn't mean you should stop using the data or trying to use the data and draw conclusions with the limitations that come with it. So it's just at most, and I always claim, I'm not gonna eliminate or replace any researcher. All I hope to do is to raise some flags that they can follow and then investigate further. That's all that we can claim that we'll ever do with automatic methods of any kind, AI, ML, and NLP.
Can I ask a question?
Sure.
And then you can have the last one while I'm hobbling up to introduce Keith. You mentioned that the Twitter API is different from the web interface.
Yes.
And I don't do this type of research, so I didn't know that. I'm wondering, is that a technical reality that you would not be able to do the pattern matching the same, or was it a philosophical decision at Twitter that the
From
programmers developed the API one way and the web interface a different way, or could they be the same? Because it seems like for what you're doing, you'd want to be able to do those patterns through the API, and I'm shocked that you can't.
I think it's a mix for Twitter. I cannot claim to be in the minds of the Twitter stakeholders. But basically, the type of queries with the patterns will be much more expensive than the keyword-based ones. So if you're collecting on keyword, it's much faster to give it to you. So everybody doing that will like bring down their systems. Plus, they sell the data. So they sell tweet collections. So.
Okay, I was interested in the heterogeneity of the people that you're following, and I could imagine that some people would be much more prone to exaggerate something compared to other people. So there's an extra sort of fuzz put on some of the data that you would be obtaining. Is there any way? Sorry, is there any way that you can use all of their other information to get some sort of a pattern for a person, so you can do some normalization of that for the specific information that you're working with?
We did some of that. So, for example, we stratified our analysis based on the total number of tweets in the timeline. So people more prolific in Twitter will tend to say more things. So, for example, frequencies of drug intake, we cannot go with just the number. We will need to know on average how many, which group of Twitter users that we're looking at are we talking about? The ones that hardly ever tweet on the ones that tweet a lot. So, so that we did in the study in that paper. So that's one way, and there's other characteristics, but we did study those characteristics and then saw the differences. Was there any difference we got country of residence, we got race, which was surprisingly easy to get, as easy can be. But we did get it, so only 14% of our timelines, we couldn't determine race, for example. And so all that data is in the paper, some of that. Okay. Thank you.
Thank you. Let's thank Graciela one more time. Okay, our final Terry talk for this year is going to be given by Keith Dunker. He is the director of the Center for Computational Biology and Bioinformatics and a professor of biochemistry and molecular biology, as well as a professor in the School of Informatics at Indiana University. He's also one of the co-organizers of PSB and has been the one leading the NIH grant effort that we've had so that we can fund trainees. So we're very grateful for that. So today he's going to talk to us about his favorite topic, which is intrinsically disordered proteins. So thank you, Keith.
Thank you very much. Okay. I presume everyone can hear me. This sounds pretty loud. Maybe I should move it down a bit. Is that so? Hello? Hello? Okay. First, I'm going to start with a question. How many in here have taken a biochemistry class? Okay. In those classes, how many gave 2 lectures on disordered proteins? 1 lecture? 0 lectures. Okay. So I'm going to talk about a topic that should be covered, in my opinion, but is not. And in Marilyn's talk, she said you should make no more than 3 points. I'm going to make 1 point. In sequence to intrinsically disordered protein ensemble to function is for signaling. Okay, that's the point. You can go. All right. So what our biochemistry textbooks contain are many, many, many examples that follow this paradigm. Amino acid sequence folds into 3-dimensional structure, and 3-dimensional structure is necessary for protein function, either by lock and key or induced fit. And this is the paradigm that's found in textbooks throughout. By virtually every protein that's discussed fits this idea. However, some proteins and regions lack 3-dimensional structure. We call these intrinsically disordered proteins. To give a very brief history, the idea that a protein could be unfolded and then inside the cell and then bind to different partners by changing its shape because it's unfolded and flexible, that was first proposed in 1939 by Carl Landsteiner, who incidentally got the Nobel Prize, and not for this, but for other work. And Linus Pauling in 1940 followed up Landsteiner's work and wrote a beautiful paper on this same idea. And of course, everyone's heard of Linus Pauling. And experiments in the 50s demonstrated the actual existence of such proteins in the laboratory. And many examples, maybe as many as 100 examples, were found between the 60s to 90s. But they've been ignored. They've been ignored. Your data of how many of you guys have heard about these or had lectures on these as a demonstration of this ignoring these guys. Okay. And I think they've not been in textbooks because they don't fit the sequence, the structure to function paradigm. They violate that. And now they are beginning to be in textbooks now. And the key event that happened that changed many, many examples scattered in the literature to the idea that these proteins exist and they're functional have been the use of bioinformatics to study this class of proteins. That was a key event to turn hundreds of isolated examples that were completely ignored into what's now a vibrant field of study in biochemistry. Okay? Our definition of intrinsically disordered proteins is that whole proteins and regions are intrinsically disordered if they lack a stable 3D structure and they're flexible molecules, that is, they're dynamic ensembles. Okay, so here's the new idea. In IDP structure function, amino acid sequence, protein non-folding, not folding, goes into an IDP ensemble, and this has actually been demonstrated by NMR experiments of proteins inside of living cells. So there's no doubt that they exist, whether or not they exist in the cell, and giving protein function. And the fundamental widespread importance of these proteins, which I'll demonstrate in a few minutes, has been shown by computational biology. And now the leading biochemistry textbooks have maybe 1 or 2 pages to introduce these proteins. So that's at least a start. Okay. Why don't they fold? Amino acid composition determines whether a protein will fold or remain unfolded. For compositions that favor folding, that favor structure, the sequence patterns of hydrophobics and hydrophilics determines which 3-dimensional fold they'll go into. But the key here, and this was in a paper published in 1993, the key idea is if there are not enough hydrophobics, a protein just simply can't fold. That's the key idea. Now, in this paper, they did not propose that there were in nature non-folding proteins. They were discussing folding from a theoretical point of view, and from the theoretical point of view, hydrophobics driving protein folding has been an idea since the 1940s, and this paper was a further elaboration of those ideas. Okay, so here's an experiment we did. We collected from the literature proteins of 20 regions of proteins of length 21 amino acids, or of 21 amino acids. We had a large collection on the order of a couple hundred that were structured and a couple hundred that were disordered. Then we partitioned them. Then we partition them. If this region of 21 has 0 aromatics, that is phenylalanine, tryptophan, tyrosine, if they had 0 aromatics, 80% of them were unstructured in red, almost 80%, and more or less 20% were structured. If they had 1 aromatic, it was about even, 2 aromatics, 3 aromatics. By 4 aromatics, more than 80% were structured in something. Now, in this experiment, this aromatics ranked 6 out of 38 different combinations of amino acids that we did in this experiment. And so, what did we find in this experiment? That if there are too few aromatics, they wouldn't fold. If the ratio of hydrophilic amino acids to hydrophobic amino acids was too high, the proteins wouldn't fold. If they had a low sequence complexity, that is, a fewer number of amino acids, they wouldn't fold. If they have a net charge repulsion would prevent folding, they wouldn't fold. If they have too many prolines, and remember, a proline amino acid does not have an NH group. If it doesn't have an NH group, it can't form a helix, it can't form a sheet. If they have too many prolines, they can't fold. So the bottom line here is the amino acid compositions of these proteins and regions is such that the proteins don't fold by themselves. So we use standard machine learning techniques to develop a predictor. In this group, I don't have to elaborate. And so now I'm going to show some of the results that we found. So there are things called signaling domains. SH2, SH3 are 2 of the most famous ones. Here's one. called GYF domain. It has, I'll get this eventually. It has this motif in the GYF domain. And anyway, let's just move on. So we see here's 100 or so different signaling domains. Here's the GYF domain in green. Now, if you look at the disorder predictor in the bottom left, bless it. Which one is the, here's. If you look at the protein containing the GYF domain in disorder prediction here, up is disordered, down is structured. The GYF domain is a structured domain in a mostly disordered protein, but it binds to it binds to the CD2, which is a cluster of differentiation protein. And you'll notice this is a transmembrane protein. This is exterior to the membrane. It's predicted to be structured. Transmembrane region predicted to be structured. And then the internal region that is predicted to be disordered. Now, it turns out that most, if not all, transmembrane proteins that have a single pass membrane protein, almost all of those proteins have disordered domains on the interior of the cell. And you'll notice a little subsegment of this gives you the binding site for the GYF domain. All right? So here's an idea. If you go into Protein Data Bank, you can find hundreds of examples, or in fact thousands of examples, of short peptides bound to globular proteins. We took hundreds of those, mapped them back to the full-length sequence, did order-disorder prediction, and nearly all of those peptides, in fact, every one that we've looked at so far, are mapped to regions that are predicted to be disordered. So if you go into PDB, Protein Data Bank, and you see a peptide bound to a globular protein, that peptide almost certainly came from a disordered region of protein, okay? Now, what are these peptide-protein interactions used for? They're used for signaling. So that comes back to the point I'm trying to make. Disorder is for signaling. So I could really say I'm trying to make 2 points here. Disorder is for signaling, and there's lots of data to support it. You don't have to remember the details, but you're going to see lots of examples. Okay. So here's another protein. I think, how many people have heard of P53? Yeah, okay, there we go. How many know that disorder is important for P53 function? Okay, now you all do, okay? So P53, if you look at the disorder prediction, and this prediction has been verified by experiment, down is structure, here it is, the central region is structured. This central domain, which is structured, is a DNA binding domain. There it is. The amino terminal region is disordered. The carboxy terminal region is disordered. This binds DNA. What does this do? It binds to multiple different partners. This little segment binds to this protein. This little segment binds to that protein. So one region of disorder can bind to 30, 40, 50 different partners. Why? By changing its shape. How? That's how it does it. And that's what we're seeing here. I mean, if you look at this little segment right here, when it's bound to one partner, it's a random coil. When it's bound to another partner, it's a beta sheet. When it's bound to another partner, it's an alpha helix. So it changes its shape. This goes back to the Pauling proposal in 1940, that unstructured regions can change their shape and bind to different partners. So we've just verified his 1940 proposal, okay? Now, what else do I want to say about this? That's enough. But, the other thing I want to say about this, if you take a protein and predict disorder, and it has structure in some part and disorder in the other parts, those disordered parts will very often contain binding sites for partners. And if you've ever seen these protein-protein interaction networks, where there'll be so-called hub proteins, and these hub proteins bind to hundreds of partners in the protein interaction networks, that ability to bind to hundreds of partners comes about because of disorder. And it's one of two ways. The disordered region itself can bind to 100 partners, or a structured region can bind to 100 partners that use disorder to bind to that structured region. Okay. I don't think I'm keeping up with the schedule here. I better go faster. Okay, here's another major point. We did disorder prediction on a bunch of viruses, a bunch of archaeobacteria, common bacteria, single-cell eukaryotes, multicellular eukaryotes. This is the proteome size. This is the percentage of amino acids that are disordered. The only thing I want you to take out of this slide is right here. Humans, us, we in this room, on the order of 40 to 50% of our amino acids are located in disordered regions. Okay? So maybe 2 points of this lecture. 40 to 50% of our amino acids are in disordered regions. Okay? But they're not discussed in your biochemistry textbook. That's a crime, in my opinion. Okay, so now, what about their overall global function? We've already seen one function, they bind to protein partners. Okay, so we collect Swiss protein function-specific sequences, or now uniprot, but at the time we did this, it was Swiss protein. We collect matching random function sequences. We repeat this 1,000 times, so we have 1,000 matching function sequences. We predict disorder on each function-specific sequence and 1,000 random function sets. And the random function sets more or less are a Gaussian, so we can rank the structure and disorder-associated functions by their z-scores, right? Everyone in here knows what a z-score is. Okay, so if we look at the top 10 biological processes most strongly associated with low prediction of disorder, what are they? Biosynthesis, transport, biosynthesis, catabolism. So what are these? These high-level functions are either transport across the membrane or something that's carried out by enzymes. We know enzymes are structured. We won't go into that, but they have to be. And we know that membrane proteins have to be structured in the low dielectric of the membrane. So these are carried out by structured proteins. Fine. Makes sense. Now, the top 10 biological processes most strongly associated with high prediction of disorder. Number 1, differentiation. Multicellular organisms would not exist were it not for intrinsically disordered proteins. Why? Because multicellular organism, for one cell type to change into 2 cell types, what do you need? You need signaling. First of all, you need to turn some genes on, turn other genes off. How's that accomplished? That's accomplished by transcription factors. What about transcription factors? Transcription factors in eukaryotic cells are massively disordered, and that enables them to differentially regulate different genes. That's number 1. Okay? Number 2, that's number 1. That's good enough. Okay. Now, so here we have 2 structure-function paradigms. Sequence to structure to function. This is for catalysis, for transport, for binding to molecules like hemoglobin, I didn't talk about that, binding to macromolecules, and binding to IDP regions. Sequence to IDP ensembles to function are for signaling, regulation, recognition, and control. How much time do I have? Where am I?
6 more minutes.
Okay. I don't know if I'll make it, but we'll get close. Okay, so we're going to go through one signaling pathway to look at this. Okay? This is a pathway, say, for T cells. There's a protein called calmodulin. That binds calcium. There's a protein called calcineurin. That's a phosphatase. There's a protein called nuclear factor of activated T cells. So basically, calcium comes in, calcium binds to calmodulin, calmodulin binds to calcineurin. That turns it on. It takes a phosphate off of NFAT, actually many phosphates off of NFAT. So then NFAT goes into the nucleus, turns on genes, and away we go. So here is calcineurin and calmodulin. Calmodulin wraps around the target, okay? So that target has to be in a piece of peptide that's away from the body of the protein. It's going to be disordered if it's away from the body of the protein. Where is that target? That target is in 100 and something residue disordered region in that protein. And that target has two different factors. It has what's called an autoinhibitory peptide that binds to the active site and turns calcineurin off, and then it has a binding site for calmodulin. When calmodulin binds, it turns it on. So basically, this long disordered region is an on-off switch that connects calcium signaling with phosphorylation, dephosphorylation signaling. These are the two of the most widely used signaling systems in eukaryotic cells, right? And so the connector between these two is a region of disorder. Okay, we'll skip this. I've made these points already. Now, what about the nuclear factor of activated T cells? I'll skip this. The nuclear factor of activated T cells has a structured region. This is pretty long. It's not emphasized right here. 428 to 697. So it's a couple hundred residues long. And that's structured, and that's a DNA binding domain. Remember P53, the transcription factor that had a structured DNA binding domain and flanking disordered regions. This likewise has a structured DNA binding domain and flanking regions. Here is the nuclear localization signal to enable it to get into the nucleus. It also has all these red dots are phosphates. So it's not one phosphate, it's a whole bunch of them. It has a binding site to calcineurin. This is all disordered and flexible. So that one binding site can bind to calcineurin, and then by the flexibility, the calcineurin can take one phosphate off after another. Then that frees up the nuclear localization signal. Then the nuclear localization signal becomes active, and it goes into the nucleus, and it does its thing. Okay? So this signaling pathway has calcineurin, which has a large disordered region, which we talked about. It has NFAT, which has a large disordered region, which we talked about. We didn't talk about calmodulin, but it has two calcium binding domains connected by a flexible linker that wrap around. Okay? So all three of these proteins have massive amounts of disorder. Okay? In summary, we have examined structure disorder for many signaling pathways, such as Wnt, Notch, Hedgehog, JAK-STAT, nuclear hormone receptor signaling pathways, NF-kappa B, and so on. All of these signaling pathways massively use disorder like the signaling pathway you just saw. Okay? And so, in conclusion, sequence to IDP ensemble to function is by disorder, is for signaling. Okay? And I'll skip this slide to keep the time, and thank you very much. Now, why aren't you taught about these proteins in your courses? I don't understand.
That was going to be my question.
Yeah. Well, if that's your question, I'll give my impression. The ideas of structure being important for chemistry started in the 1850s with the whole idea of the structure of benzene, so-called like a snake biting its tail. And from 1850 until 1950, at least, the whole thinking about chemistry and biochemistry was dominated by thinking about structure. Disorder depends hugely on entropy, and that's a concept nobody understands. Okay, let's go.
Okay, so thank you. That was very stimulating. Two questions about what you would consider disordered regions. So first one is that I would guess there'd be some correlation when you're looking at hydrophilic regions on a protein. A lot of those would tend to be on the outside of the protein. If you're looking for interaction sites, those would also often be on the outside of the protein. So I'd imagine there'd be some correlation with what you're identifying as disordered regions versus regions that are on the surface.
Okay,
And
can I answer that?
Sure.
Okay, so what happens for the regions that are on surface of proteins, yes, they have hydrophilics on the surface, but the next residue will be a hydrophobic to push it down to attach. So you need a balance of hydrophobics and hydrophilics with a pattern such that you can slap it down and have it structured, but have the hydrophilics pointing out. The disordered region will have lots of hydrophilics in a row, and so it doesn't plaster down, but it will have an occasional hydrophobic here and there, so it can bind to a templated place where those hydrophobics will fit into specific spots. So they can bind to a surface, but they can't fold by themselves.
Okay.
I hope that answers your question.
yes, no, that's interesting. So the other issue I was wondering about was a lot of these proteins are multi-subunit. So
Are
when
what?
They have multiple subunits.
Yes.
So if you look at an individual subunit, there may be a region which in isolation is disordered, but once it's assembled into the final complex, then it's stabilized by other components.
Yeah.
So in that case, would you consider that a disordered region or not?
Well, we can actually, Ruth Nusinoff and a postdoc, I can't quite remember his name, developed a mechanism for distinguishing what you're talking about versus globular proteins. So if I have two globular proteins and they bind together, what happens is they have a small surface area because they're like a sphere, and they have a small interfacial area because the two spheres coming together have a small interfacial area. If I have a protein that's fundamentally disordered and another protein or another region of the same protein or another protein that's fundamentally disordered, they bind and they intertwine. So if I pull them apart by computer, you'll have a large surface area of the monomer, you'll have a large surface area of the interaction. And so if you plot monomer surface area versus interaction surface area, structured proteins are here, disordered proteins are up here. And so it turns out the protein data bank is loaded with these kinds of structures, okay? And I would still call them disordered because when they're by themselves, they would be disordered, and they only form structure in the presence of their partners.
Right, but the assembly of those units is often going along also almost during translation. So
Oh, yeah. Well, I don't know about during translation, but it happens soon and it's permanent, okay? So, but it's still you're asking my opinion.
Yes.
And in my opinion, I would say that the monomers are disordered,
Right.
even if it's only for a microsecond before they assemble with something else.
Yes. So it's a mechanism. In
They
that
don't
case to
fold first and
help
bind
assemble.
second. They bind and fold at the same time. That would be my distinction. Okay.
Keith, thank you for the fabulous talk. One of the things I was wondering about is different disordered regions can interact with different ligands, not just proteins. And so I was wondering if you've been able to learn models for what disordered regions are going to interact with what different kind of ligand, and beyond that, what the specificity of that interaction may be.
I haven't done that. Other people are working on that and done a much better job, and I'm not really competent to talk about their results. But I know that, for example, I'll give one example. How many here have heard about the Lac Repressor, right? This is the classic signaling protein. This was the first one that was figured out by Jacob and Monod, and they got the Nobel Prize for that. That has a tetramerization domain, that has a headpiece that binds DNA, and it has a 12 residue disordered linker. And that 12 residue disordered linker is absolutely crucial for its function and becomes involved in the binding to the DNA. So you can find lots of examples where it's going on, but I'm not an expert in all of those. So.
Thanks for a talk on something that's near and dear to my heart as well. So for designing drugs, we know
Pardon?
that designing drugs, pharmaceuticals, crystal structures are generally, we kind of weigh those a lot.
Yeah.
Use them a lot, and IDPs can't be crystallized because they're disordered. So I was curious if there's some good examples of pharmaceuticals that have been designed based on IDPs. If there haven't, what would you propose as kind of a pipeline to do that?
Okay, I'm taking a drug which acts on a disordered protein. The drug, however, doesn't bind to the disordered protein. It binds to the target of the disordered protein. Okay, angiotensin II is an unstructured peptide, and it binds to the angiotensin II receptor, which is a GPCR, a G protein coupled receptor. And the drug that I take is valsartan, is a generic name, and it binds to the pocket that is the target of the disordered region. And it's my opinion that over time, we will find lots of drugs binding to targets that disordered proteins bind to. And there will be a huge number of drugs along this line. Why? Because it depends on the same principles of drug design that we already know about. It's just the targets are different. That's the first thing. Other people have found small molecules that actually bind to disordered regions and prevent them from binding to other disordered regions. There's a very famous one where there's a complex that's between a protein called c-MYC and another protein called MAX. And 6 or 7 different small molecules have been found to interrupt the MYC-MAX complex formation, which is important in a lot of cancers. Okay, now, the problem is it doesn't bind tightly enough. And so there were several of them. NMR showed that they all bound to c-MYC and not to MAX. And so they strung them together by linking them, and they got nanomolar binding to c-MYC, right? We're going to have a drug, lethal as heck. It's very toxic, okay? And so, I mean, it's really huge and does a great job of killing the animals, right? And so I'm still hopeful that these guys who are much smarter than I am are going to figure out how to do this. But the big problem with drugs that bind the disordered regions is really weak binding because they're floppy and you need a large drug to have enough surface area to overcome the entropy of the flexible chain. And to find a large drug that isn't toxic is really difficult. Okay?
I'm sorry to cut
I
off the
couldn't be more
questions,
hopeful.
but we're
Pardon?
dipping into the next session, so maybe the rest of you can find Keith afterwards. So let's thank him one more time for a great talk. And I'm going to pass it over to Lana to run the next session.